Hello my friends, this is a quick revision on the molecular biology practical curriculum. First, we will start with DNA extraction. As we know, DNA is located inside a nucleus and the nucleus is located inside a cell. So if we want to take DNA out of nucleus, out of cell, we should first dissolve the barriers in our ways in the form of phospholipid bilayers. We can do this by mechanical crushing, and we can do this by exposing these compartments to soup, which is a dissolvent for the lipophilic structures. After we dissolve these barriers, so DNA most probably out of cell, but is mixed with other proteins and other ions, other enzymes that can destruct the DNA. So we need to add to soup other compartments in order to guarantee the extraction and guarantee the purification. We can add NaCl. NaCl has a positive charge on Na and negative charge on Cl. So the positively charged sodium will bind to the negatively charged DNA. The negative charged DNA source is the phosphate group, the free phosphate group at the 5' end. And the chloride in negative charge will bind to the other proteins that are positively charged. And I mean here the closest to DNA histone protein. It has the priority to be extracted out of DNA. There are other destructing enzymes. So in order to abolish their effect on DNA, we should add editor. Editor act as chelators for magnesium, for calcium, ions that are necessary for the action of such destructing enzymes. So we extract DNA, we separate it from the closest proteins, histones, and we break down the action of the breaking or destructing enzymes. The three of these components in NaCl, editor, and soup is called extraction buffer. So DNA is out and somehow purified. How can we show up, display DNA? This is done by adding 90% ice cold alcohol. Alcohol has a partial negative charge, okay, that can bind to the partial positive charge on H2O. H2O by positive charge can bind to the phosphate, the free phosphate group at the 5' end. So by adding alcohol, the partial negative charge on alcohol bind with the partial positive charge on water and prevent such binding from being existent. So the negatively charged phosphate group bind to Na positively charged completely. So we prevent the dissolvent or the dissolution of DNA in water and make DNA precipitate at the water alcohol interface. There is a minor trick here. Alcohol should be added slowly and on the wall in order not to disturb the mix and in order to make DNA appear at the interface quickly. So in summary, the materials required, DNA source, tomato slices, extraction buffer, as we said, 95 to 100 or 90% ethyl alcohol, knives for mechanical crushing, watch glass for putting these materials or the specifically the tomato slices on, nylon gauze to filtrate the juice from the pieces, funnel, to add such compartments, or to add the juice, to pour the juice inside the test tube. The steps: mechanical crushing, the extraction buffer, nylon gauze and funnel to get fibers free solution, and pour equal volume of ice alcohol. Observe the layer, whitish cotton-like substance appears at the interface, and this is the DNA. Okay, the measures 2 milliliter of extraction buffer plus 2 milliliter and equal volume of alcohol. So we have DNA extracted. We want to make sure this is DNA first, okay? And we want to measure or to determine if it is purified or not, by making sure it is DNA by a substance called diphenylamine. This diphenylamine can bind to the deoxyribose structure on the double-stranded DNA. When it binds to deoxyribose, it gives a bluish color. So the more we have bluish color, the more we have DNA. The other question, whether DNA is purified or not, by checking for the presence of the nitrogenous bases. This is done by using UV spectrophotometer at 260 nanometer. This is the wavelength to be selected for determining or for measuring DNA inside the solution. To check if this DNA is purified from proteins, we should measure the absorbance of the predicted or the supposedly present proteins spoiling the sample at 280 nanometer. So 80, 84 protein. By dividing the absorbance at 260 nanometer, that belongs to DNA, over the absorbance at 280 nanometer, that belongs to protein. So the more we have the value, the more we have DNA inside the solution. They calculate it and they determine an acceptable ratio between 1.6 and 2. If the calculus or the result is inside this range, so we can determine, we can say this DNA is purified enough to proceed to the next procedures. There is a word or a term here called hyperchromicity. Hyperchromicity means if we heat this double-stranded DNA, then we unspool to dismantle the double-stranded DNA to single-stranded DNA. The definition of denaturation, if you are American, or denaturation if you are British. So we have deoxyribose, more deoxyribose in the exposed in the sample by heating. We have more nitrogenous bases exposed by heating, so that the measures of absorbance will be increased, and the intensity of the color, the absorbance of the diphenylamine, or the product of diphenylamine, will also be increased by heating to 95 Celsius. So this was for DNA extraction. Let's shift gear to electrophoresis. Electrophysis to separate DNA fragments according to their molecular weight. And we exemplify this procedure by this man who is supposed to, throw this ball to as far as he can. If this man is strong enough, the ball will migrate far away. If the ball is small or of small size, of a small weight, likely it will migrate far away too. So the factors depend, or agarose gel electrophoresis depend on the electric current power. Okay, this is exemplified by the man who's supposed to throw the ball. The molecular weight, as we said, of the ball, and I mean here in our case of DNA, the molecular weight of DNA fragments, and the medium, right? The medium, because if we have a wind in the air, this ball can be swayed from its way, can be stopped. Okay, so the medium plays a role in the velocity or in the rate of migration. The medium here is agarose gel in the case of DNA. So as you see in this slide, larger pieces is close to the stage, which is the cathode, because DNA fragments are supposed to be negatively charged here, negative charge DNA. They are close to the stage. This is the stage, the cathode, and they are supposed to migrate this way to reach the anode, okay, or to be repulsed from the cathode. So the larger the sizes, the closer they will be near the stage. You can see here the smallest fragment. You can see here other smaller fragments as we go toward the anode. To set up the electrophoretic chamber, so we need the medium, the agarose gel. So we should have custom tray for the agarose gel to pour it inside this tray. After it will be cooled, it will be appropriate for DNA to migrate through, but we should make pores, or should make holes, I mean, for DNA samples. We can do this by what's called gene combs. We insert gene combs here in this place before gel is cooled, so that after it's cooled we have holes, we have wells for DNA samples. Then we prepare the buffer with a specific pH, with a specific electrolyte component. The buffer, you can see the liquid here, right? First, the buffer is used with DNA, with gel for preparation, and the buffer can be poured for the gel to be soaked into, so that it can facilitate electrolyte movement. It's a good conductor for electricity. We have gel tank for the gel, so the custom tray with gel, okay, will be put here, will sit up here, soaked inside the buffer, like we see in this photo. Casting tray, buffer, the cathode is here, the anode will be here. So we now we have to conduct the electricity inside such chamber, and this is by electrical leads that will cover the whole chamber this way, and we put into an electric source, so the electricity passes, and DNA by its negative charge can be repulsed, can be forced to move and migrate this way toward the anode. But I bet you noticed that DNA are cut into pieces, right? DNA fragments, or DNA's the genetic makeup is cut into pieces. How is that? How can it happen, or how can we cut DNA fragments into such pieces? And this is by restriction enzymes. What are restriction enzymes? They are enzymes produced from bacteria originally to stop the invasion of viruses, other viruses. Restriction enzymes cut the RNA of viruses at site called palindromic site, at which the sequence, the order of nucleotides from 5' to 3' are the same on the opposite direction. You can see here GAA and GAA. Here from 5' to 3', this strand, and 5', 3', the other opposite strand. Okay? So restriction enzymes produced from bacteria to cut RNA fragments at palindromic sites. How can restriction enzymes distinguish between bacteria and viral genetic material? By methylation. Okay, methylation. The bacterial DNA is methylated, so restriction enzymes can figure out this genetic or this DNA belongs to bacteria and cannot destruct it. Okay, and the other viral genetic material is not methylated, so restriction enzymes can work on it and cut these RNA fragments and prevent viruses from invading such bacteria. We can make use of restriction enzymes by cutting the DNA into fragments of different molecular weight. Okay, so the larger will be here, and the smallest, the larger will be closer to the stage, and the smaller will be closer to the anode. How to visualize? We have two stages at which we need to visualize DNA. First, during the run, and after the run to interpret our results. During the run, by tracking dye called bromophenol blue. Bromophenol blue, from its name, it's blue, and it's mixed with DNA, so that when DNA migrates in this way, we can see the migration. We can see the dye is transmitted or transferred from this position to this position, so we can make sure that DNA migrates well. After the run, we have an ethidium bromide dye, and this dye is teratogenic and toxic, so we should take care by wearing gloves and so on, other precaution. And the interpretation is examined or is by UV ultraviolet rays using what's called ultraviolet trans illuminator. This is an example of what you can see as gel electrophysis product. We can see here a piece of DNA at a specific size, of a specific quantity, and I'll tell you how we can figure out quantity as well as size. This is DNA ladder, a preset DNA fragments. We properly know their sizes. Okay, so we have here 5,000 base pair, as we can see it will be here, 2,000 base pair. Here we have 100 base pair in this place. So this is the DNA ladder ordered from larger sizes above to smaller sizes above. So if we have a specific segment that stops here after agarose gel electrophysis, by examining the number at the ladder, so we can determine this piece equals 500 base pair. This is for molecular weight. For molecular or the quantity, the more intense the band, like we see here in lane 1. This is called lane 1, lane 2, lane 3, 4. As we see in lane 1, the more intense is the band, the more quantity it has. Here we can see a faint band, so the quantity is lower than the quantity here. Okay, and here we have more quantity. So DNA extraction, electrophysis. Let's shift gear to discuss PCR, polymerase chain reaction. As we can see here, the stars can be magnified by telescopes. The magnification is in sizes, right? So we can determine their actual sizes. We can determine or can see the details of such stars. By using polymerase chain reaction, we can do the same, but not magnification in size, but magnification in quantity. Okay, gene copy numbers can increase the gene copy number in an amplification technique. There are another amplification technique called molecular gene cloning. This one is by using bacterial replicative machinery to amplify genes in numbers. But here in PCR, as you see, the whole reaction occurs inside a test tube. It occurs in vitro. So we don't have sources, we don't have supplies for replication, so we need to supply this reaction ourselves. How can we do this? Let's assume we will mimic the DNA replication. We'll build DNA components, other strand complementary to the strand we have. So we need to have DNA source at which it lies the gene we need to amplify, and we need the brackets, nucleotides in the form of synthetic nucleotides. We need to the man or the signal that determines our the start of our reaction, DNA primer, and we need the workers, which is Taq polymerase. Taq polymerase differs from the DNA polymerase inside our body that catalyze the DNA replication in vivo, but here we are working in vitro, and we will modulate the temperature. So we need an enzyme that can resist denaturation with changes in temperature, and it can work optimally at a higher temperature that can reach 72 Celsius. Taq polymerase, the abbreviation T from Thermophilus and AQ from Aquaticus. It derives from the bacteria that favors living in high temperature aquatic region. So this is for Taq polymerase. How is the chain reaction maintained? The first step, we do denaturation to separate, to unspools the double-stranded DNA from each other and make the gene exposed like here. Okay. By annealing, we make the components to take the standby mode. The DNA primer binds to its complementary specific site. The nucleotides are freely floating here in the test tube, and the Taq polymerase will be here ready for doing its job. Once we raise the temperature to the optimal temperature 72, the Taq polymerase catalyzes the reaction and uses the free-floating nucleotides to add in a complementary manner to the template strand containing the gene of interest. So the keyword here: temperature, denaturation to separate, annealing to make the components in a standby mode, and elongation for the ongoing process. By the repetition of the last cycle, denaturation, annealing, and elongation, we can get more copies of DNA. For example, if the first cycle produces 2 copies of the wanted gene, then if we repeat the cycle again, if we repeat the polymerase chain reaction again with 2 genes, we can reach 4 copies. With 4 copies, we can reach 8 copies at the third cycle. If we repeat it another cycle, 4th cycle, then we get 16 copies. So they tell us a rule, a formula: 2 to the power of n. N refers to number of cycle, provided that you start with one copy of gene. Okay, so we start here with 1 copy. If he is asking you after 30 or after 35 cycle, how many copies can you get? Simply by squaring 2 to the number of cycle, here 35, so we can get 34 billion copies. This pattern of amplification is called exponential amplification, where 2 will be 4, will be 8, will be 16, not 2, 4, 6, 8, 10. You can see this is exponential. This will accumulate larger copies in smaller times. If we want to do PCR but not for DNA, we need to amplify a gene copy on RNA. First, we should reverse RNA to DNA, complementary DNA, okay? And then we repeat, or we do the same cycle and repeat the cycle again and again till we reach the gene copy. It's called real-time PCR, or by using reverse transcriptase enzyme for the transversion process RNA to DNA. So it's the same, but by using reverse transcriptase first, and you can see more details in the theoretical curriculum. So DNA extraction, electrophysis, BCR. For blotting, it's like the old camera, the old photo generation. You can see here the Kodak film. For the old cameras, the film was like this, and this wasn't the photo you got 10 years ago, but they blot this photo inside a white copy, so that you can get your photos in colorful manner. We can do the same after electrophysis. We get the gel after agarose gel electrophysis, DNA fragments separated by sizes, but we need to do blotting. Why? Because we want to add chemicals for other probing procedures. We want to detect targets. We want to do fingerprinting, other procedures that requires applying chemicals, and the agarose gel is not satisfactory, or is not suitable for such a chemical modification. So we need first to transfer fragments to a more compatible filter, a more compatible membrane, okay, for the chemicals we will use for probing and for detecting targets. The same fashion with the camera. This is the gel. This is the nitrocellulose filter. We want to blot the fragments on it. We need to make the gel tightly adherent with nitrocellulose filter by adding paper towels and adding weight over the paper towels, so that we can make sure they are tightly adherent. And we will put the gel with the nitrocellulose inside the buffer. The fragments will move upward to be blotted on the nitrocellulose membrane. We get the nitrocellulose filter with DNA fragments. Now we can do the probing, the hybridization, the targeting procedure after. How can we do this? We want to transfer this faint photo to this shiny outcome. We have types of blotting for DNA. If we start with DNA, then we have southern, not southern. Okay, this is the name of the scientist. And for RNA, if we want to plot RNA and find out targets on RNA, the technique we will use is northern. You can see R with R, RNA northern. Protein with T, we have western with T for proteins. Let's suppose the target is here. The target is here. This is the target. So we will manufacture what's called a probe. If this target is RNA or DNA, so the RNA and DNA contain oligonucleotides. So the probe we will use will be single-stranded oligonucleotides that can bind specifically to this RNA, to this DNA, in what we call molecular hybridization. Okay, if we have here protein, then protein is a large and is amino acid sequence, not nucleotide sequence. So the probe will be antigen or antibody that can bind specifically to the protein we want to see it's existent or not. After we do the probing procedure, we need to we need to visualize such binding in the case of DNA in the molecular hybridization, or in the case of protein with antigen antibody. So we will use either radioactive material, phosphate 32, high molecular weight phosphate, or fluorescent dye. So this shiny pattern will display in a nice photo like this. You can see, yes, there is a target in the sample. Okay. Let's wrap up the uses and clinical applications of all of the procedures we have discussed in the molecular biology practical curriculum. DNA extraction, this is for preparation and purification of genetic materials for next procedures, and we have discussed how and why. Okay. Electrophysis, to separate molecular sizes, different molecular sizes of DNA, and to set up the quantity and to determine the quantity. PCR for diagnosis of infectious pathogen that take a long time, like tuberculosis, and that are complicated, way too complicated if we do it the ordinary way, like in viruses. Okay, viruses like HBV and HCV, but HBV, it is DNA, so it will we will do a conventional PCR, ordinary PCR for hepatitis B, but hepatitis C we have RNA, so we need to do RT-PCR. For blotting, we do blotting for target identification, DNA by southern, RNA northern, and protein western. Another example for blotting, DNA fingerprinting. The story begins with the human genome, which is 3 multiplied by 10 to the power of 9 base pairs. Human differs only 0.1 differences in the intergenic regions and in the tandem repeat regions. From single nucleotide polymorphism to short satellites from 2 to 9 nucleotide difference, from 10 to 100 varied numbered satellites, varied numbered tandem repeats. This is the restriction fragment length polymorphism. The differences creates or makes, if we apply restriction enzymes on these DNAs from different people, it will create different sizes if we run them on gel electrophysis, depending, as I said, on the differences in the oligonucleotide or in the nucleotide order, in the nucleotide sequence on such DNAs. Restriction enzymes, we have discussed this before. This is used, of course, for personal identification, like I said, or like you see here. Every person has its own unique pattern of DNA fragments. If we cut his DNA, if we cut his genome using such restriction enzymes, to compare these patterns in paternity tests, to compare the the evidences selected or collected in crime scenes, to determine if this evidence belongs to this person or not. Uh, and for diagnosis of hereditary or genetic disorders by adding probes specific for the mutant gene we are searching for, the same way we have done it with southern, with northern, with western blotting. For example, we can see here blood stain collected or gained from crime scene, and we have suspect 1, suspect 2, suspect 3. By doing a simple alignment with the DNA fragments here, you can see that the blood stain belongs to suspect 3, because we have here this fragment belongs to or at the same level with this one. We can see this, belongs to the same distance, so there is 100% match the fragments of the blood stain gained from blood stain extraction, electrophoresis, and blotting, southern blotting, with the southern blotting gained from the suspect 3. By in paternity test, we need to start with the child. The child here has 2 fragments. Okay, one of them is close to the stage, one of them is far a little from the stage. First, we need to determine which one of these fragments belong to the mother. This one, because it lies at the same distance. Okay, and second, we need to determine which father lies or has a fragment similar in sizes like the other ones belong to the child. You can see father, yes, father 1 has a segment but similar or lies at the same distance with the other fragment from the child. You can see father 2 has two larger DNA segments from the other mother, child, and father 1. So the child belongs to this mother, of course, and father 1. Bioinformatic, a quick revision. Bioinformatics, biomolecular biology, informatics, to apply computer science statistical tools to collect, to organize, and to interpret and analyze the data gained from molecular biology. Databases, we can make a different sets of databases according to the organized information in such databases. We can see databases for genes, databases for proteins, databases for transcriptomics. Okay, so database, it's an organized collection of information. If it was biological information, then we have biological databases. Goals lie in to determine a specific genes for specific diseases, so diagnosis. Okay, and this will make us understand the molecular mechanisms behind the behind illnesses, different illnesses, and in such a way we can innovate therapeutics targeting such mechanisms. Okay, and all of this advance the field of individualized medicine or personalized medicine. Can see different levels of molecular biology: DNA, RNA, protein, and metabolites. But it is not enough for us to analyze only genes, because there are other post-transcription modification here, post-translation modification here, problems that can occur at the 3D configurational or configuration, the 3D configuration of proteins. So we need to study beyond genes. We need to study RNAs. Transcriptomics comes from transcript, and proteomics and metabolomics. Genome means to understand or to set the whole genes, the whole genes. Transcriptome, the whole set of RNAs, the whole set of proteins. And we need to generate data for each of this level, okay, to integrate them together, and this will help us to analyze and understand better what's going on in health and in disease. In an example, in clinical example, osteogenesis imperfecta, we need to determine if there is a gene defected, if there is a gene mutant or not. So we can refer to the database we collected from a normal population that has the normal gene. This is from the database, okay. And we will collect the suspected gene. We need to determine if it is diseased or mutant or not, okay, and we will compare this gene to the reference gene. If it has the same sequence, so it's normal. If it has a nucleotide changed or a specific sequence deleted, or there's a frame shift mutation or so on, this will be considered diseased and mutant, okay. So the procedure will first we will do the gene, or extract the DNA containing the gene to compare it with the normal reference gene. Then we will not stop at the level of gene. We will go beyond to see the modification that happened after the gene is transcribed and when the gene is translated. So we will translate this gene into a specific sequence of amino acid to see the difference at the level of amino acid sequences, and we will by using another software, we will put this amino acid in 3D dimensions. The software needed for comparison of genes called Blotting, N for nucleotides. How can you determine there are nucleotides? By simply see the letters here, G, A, G, T, C, A. So they are letters for nucleotides, letters T, A, and G, C, DNA nucleotides. So these are gene sequences, okay. At the top you can see Homo sapiens, our species, humans, the modern human, and the name of the gene, collagen type 1 alpha 1, col1a1, okay. So if he is asking you in the exam whose species this sequence belongs to, the answer will be Homo sapiens. What is the name of the gene? The answer will be collagen type 1 alpha 1. What is the identities, or what is the similarity between the gene, this normal reference gene, and the mutant or the tested gene, okay. So we can see here 4 deleted nucleotides and 1 mismatch. So the total differences will be 5. The result will be plotted like this: 610 out of 615, 99% identical, okay. The second step in the chart, in the flow chart, we should translate the tested gene into a specific or a sequence of amino acids. The software to do this is called Electrophysis, X from expression, okay? It will portray, like you see, different possibilities of amino acid sequences that can derive from the tested gene sequence. Which sequence, which frame will you choose? The frame that that goes with this criteria, that achieves this criteria. What is the criteria for choice of frames? First, to start with methionine, start codon, to end with a stop codon, and the largest genetic message. And there's another sophisticated condition, that the one that goes prime 5 prime to 3 prime direction. So these are the condition you should consider when you choose your collect frame. This one start with methionine but not end with a stop codon. Okay, so this one will be cancelled. The frame 2 start with methionine and go without interruption to end with a stop codon. Yes, this and it goes from 5 prime to 3 prime. Then it can be the correct. Here we can see methionine stop, but it is pretty shorter than this one. And here we can see it starts with no, there's no methionine, there's no start, and there's only one line. It's only one line, and from 3 prime to 5 prime, so it will be cancelled. 3 prime, 5 prime, and there's an interruption here by stop codon, so it's definitely not the correct answer. So the correct answer here will be frame 2. We can take this frame 2 and supply the last software BLAST-P for protein amino acid sequence, to compare the amino acid sequences of the tested amino acid sequence with the normal reference amino acid. We put it in the software. The results come like this. We have 94 out of 141 identities in the 67%. You can see how much 1% of gene difference can make 70, or 1% difference can make 33% difference at the level of amino acid sequence. Okay. So here to focus on, we should not stop at the level of genes, and to move toward to see the similarities, to do alignment at the level of amino acids. By putting the resultant amino acid sequence in a 3D dimension, we can see here the normal gene. It has indentation and it has additional marks that of course stabilize the protein and make it do better functions. But we can see here the diseased amino acids configuration without those marks, without those additional stabilizing structures. So it figures now that the diseased protein in the 3D cannot maintain the normal function as the healthy one, so the osteogenesis imperfecta occurs in our example. So that was a quick wrap up for the curriculum. I hope it was helpful. Thank you.